Alright, so first things first, we're going to turn on our burner. Grab your glass slip, write your name on it with a Sharpie. Some of them have a frosted edge, so make sure you write it on there. And also, on the bottom of the slide, I'm going to draw a circle, about that big. You're going to grab your bottle that says sterile H2O. Now with these types of bottles, what you want to do is you want to open them up and ensure that when you open them you don't splash yourself or anyone else. Put them on the table, grab the top part, the lid, not the bottom part, because if you squeeze it when you open it, that's how you squirt it out. So grab the top and push down, and then with the other hand you're going to grab the lid and just pop it off. So just like that. You're going to take as little amount of water as possible. You can get a full drop on there, but you're just going to have to wait longer for it to dry off. So what I recommend you do is you start off very close to the plate, so you only end up putting like half a drop of water on there. They can see that on there. This does not have to stay sterile because we're going to be killing the bacteria on there anyways. Now, you'll have your plate, hopefully with the individual colonies on there. Keep it near the burner for your sterile environment. Heat up your loop till it's red hot, and then you go ahead and wait 30 seconds. Make sure you wait on it before touching any of the bacteria with your hot loop, otherwise you'll end up killing them. Yeah, there's going to be a lot of waiting steps for this. So, alright, so go ahead and grab 1 or 2 colonies, maybe even 3. Grab a decent amount on there. Once you have about 3 colonies, really you should only ever do 1, but for demonstration I'm doing 3. And you just rub it on the water in the circle. Everyone see that? Just spreading it out. And then again, you sterilize your loop just so that you don't have anything biohazardous rolling around on the table. And now you have to let it wait. This could take a while, so I prepared one before class. Once you're done once the water is completely or mostly dried out, you're going to pass it through the flames just twice, and make sure you're using a clothespin to hold the slide, otherwise it gets too hot and you can hurt yourself. This is still wet, but you're going to, just for demonstration purposes, you're going to wait till it's mostly dry. Just pass it through the flame twice and wait about 30 seconds, and then pass it twice again, and it'll be good to go. So again, this one I've already went ahead and did that, so I won't do it again. Now, each of you at your table should have a gram staining kit. You're going to start off with crystal violet. Again, make sure you open it the way I showed you how to, so you don't spray yourself or anything or get this on your skin, because it will stain you. And then you go ahead and put 5 drops. It doesn't have to be 5 drops, it can be less. Just make sure you cover the area where that circle is. And you let it sit for 60 seconds. We're done with the burner for now, so I'm just going to turn it off. once the 60 seconds have passed, you're going to, again using the sterile H2O, you're going to rinse it off. Again, don't put the water directly over the circle, but rather above it so that the water slides down. You're going to do that until most of it comes off. Of course, you're always going to have a little bit left on there, because there might be some gram-positive bacteria on there. And what you can do, in order to not have to air dry, is you can get some lens paper and blot the thing down. Now with blotting just means touching. You don't want to make contact with the paper and then streak. Otherwise you're going to ruin the fixation of the bacteria and you won't see anything. So just gently, if you want, you can put the finger on top as well. The bottom part. This is biohazardous because you got bacteria on there, so make sure you throw that away into your red biohazard trash. Thank you. Now for the next step, you're going to add iodine. Again, 5 drops. Or just as much as you need to cover the circle that you drew. And you're going to wait 30 seconds. Then after the 30 seconds are up, again with sterile water, just going to rinse that off. This time you don't have to blot it dry because we're immediately going to rinse it off, this time using the decolorizer. That's your ethanol and your acetone. And again, it's just same thing. This time you should see a lot more color coming off. You don't want to do it too much. I'd say about 5 seconds of washing off. And then you're going to wash off the decolorizer with water. And again, you're going to blot it dry. Now, once it's mostly dry, by now all the crystal violet should be stuck to any peptidoglycan that belongs to gram-positive bacteria. So now we're going to use the counter stain to stain any gram-negative bacteria using the safranin. We're going to put, again, as much as it takes to cover the little area where your bacteria are, and you're going to wait 20 seconds this time. So after the 20 seconds have passed, using water, you will clean this up. just go ahead and blot it dry one last time. With the blotting, don't be so heavy-handed like I'm doing here. I'm just doing this for demonstration just to get it done quickly, but you want to be more gentle, so you guys want to be blotting something like this. And again, don't be rubbing either, just blot dry. The bottom, of course, you don't have any bacteria on there, so you can dry that however you want. Then, once you're done, you just let it air dry for a little bit. After it's been air dried, you're going to take your water, put one drop on the center, and you're going to take a cover slip, put it on the slide itself using the drawbridge technique that you guys should know, and then you can look at it under the microscope. So the cover slip, you're going to put on with what's called the drawbridge technique, where you're going to place the cover slip on its side and then just let it fall after you put one drop of water onto the prepared bacteria. So, boop, just like that. And you should have no air bubbles if you do it correctly. You can place it underneath the microscope, and you can look at it under any of the objectives, but I'm going to show you guys specifically how to use this white one here, the 100 times oil, 100 times magnification oil immersion. You guys should have these immersion oil kind of droppers to use them. Take the cap off, put one drop, you only need one drop of oil on top of the slip. And this is the part where you have to be careful. You have to raise up the stage so that the oil comes into contact with the bottom of the objective, with the lens, but not the actual cover slip itself. So you're only going to be touching the oil. And you'll see you'll be able to see when it makes contact. Once you have the contact, you're only going to be using your fine focus. Don't be using the coarse focus. Otherwise, you're either going to break apart the contact, or you will break the cover, not the cover slip, the slide itself, because you'll be putting too much pressure on it. Well, after you put the oil on the slide, you cannot use that slide anymore for anything other than the oil, because the oil cannot be put on any of the other objectives, otherwise you'll ruin the lens. Now once you're done looking at it, to clean this, after you turn off your microscope, using the lens paper and not the oil, sorry, not the Kim wipes, use lens paper. You don't want to rub the bottom of the objective. All you do is you just kind of move back and forth with the lens paper, just rubbing off the excess. And if it needs any extra cleaning, we'll clean it, but for you guys, just worry about taking off the excess oil. And that's it. Any questions on that? All right. Alright, so now I'm going to demonstrate how to do the stab and swab. So you guys each should have empty plates. Make sure you write your name along the edge of it. Well, in this case it doesn't matter where you write it. Write your name on it, and you'll grab a potato slice that we have sitting in some sterile media in the back of the room. Get some tweezers or even your needle. Heat sterilize it. The needles will take a little bit longer to heat up because it's more metal. But once they get red hot, you can just go ahead and grab the potato slice. You don't have to worry about it being too hot, since you'll since it'll dry there. Go ahead and put the potato slice again in your plate. Make sure it's within the area of the fire because you're trying to keep this as sterile as possible. One thing that I forgot though is to put one of these little cotton swabs underneath it to keep the potato moist. Now remember, we want to have a conducive environment for whatever causes the disease. A high humidity is usually favorable for those pathogens that infect plants. So we want to keep this here as moist as possible. Go ahead and grab one or two of these. And just dip it in the water once. Now where I screwed up is I should have done this before I put the potato on the plate. So I gotta do that. Flip it around. And sterilize. Alright, so we're all done with this. Now we're going to do the actual inoculation itself. Take your needle and heat sterilize it. And go ahead and just stab the potato at different areas. And just sterilize it. Now at this point, you can either use your inoculating loop to pick up a colony and rub the area where you stabbed it, or you can pick up a colony with your needle and then just stab it again. Either or. Personally, I think it's faster just to pick up the colony using the needle. So make sure the needle is cooled down a bit though. Technically this isn't the stab and swab if I'm only stabbing, so I'd probably get in trouble for that. But it'll save you guys some time. And just pick up one colony after it's dry. Go ahead and stab potato and sterilize. And you can do that as many times as you want with as many different colonies or different cultures that you guys have. Make sure if you do have different cultures that you write down which is which. And make sure you also have a negative control, which is basically what I did at the beginning where all I did was stab and not do anything. But for the actual swabbing part, just so I don't get in trouble for not demonstrating it, you heat up your inoculating loop, let it cool down after about 15 seconds, or you can just touch a part of the media that's not used. Pick up one colony, find one of those first stabs that you did, and then just rub the area. And as simple as that. The bacteria will start to grow, they'll get inside, and they'll start to rot the potato. And that's it. Any questions? All right.